Protein Dynamic Solutions continues its efforts to provide the advantages of our system for the biopharma industry, in the form of a series of videos demonstrating the capabilities of our ProteinMentor platform technology. ProteinMentor is a benchtop instrument that is cloud-ready with minimal cost of operations, requiring only dry air purge. Equipped with a wide-field transmission optical setup and 4 external cavity quantum cascade lasers, which provide a highly intense broadband mid-IR light source within the 1,800 to 1,000 wavenumber spectral region. The quantum cascade lasers are transmitted through the entire field of view of a well, which has a fixed path length for an extremely fast image acquisition of the sample. A ratio of the transmitted quantum cascade laser's light source, with and without sample, is captured by a focal plane array that is 480 by 480 pixels, and yields the absorbance spectrum for each pixel within the hyperspectral image or spectral data cube. While current technologies limit their understanding by focusing solely on the region of the amide 1 band, ProteinMentor expands its view beyond the amide 1 spectral region. Its rapid method of analysis that is highly selective and sensitive, aiding in the exploration of new modalities, and provides a clear understanding of the protein dynamics in solution with direct amino acid level resolution. The system allows for thermal control flexibility to ensure that the spectral data acquired is representative of the set temperature and fit for the multiple two-dimensional correlation algorithms that may be applied. We are illustrating 4 examples to provide the conceptual framework for the thermal setup, showcasing adaptability to the customer needs. The first, a single temperature evaluation for comparability assessment against a reference standard in just 10 minutes for up to 21 samples and 2 controls. The second, a custom temperature ramp which in just 45 minutes evaluates 21 samples and 2 controls. The third, a custom temperature ramp with a limited sample array of 4 samples and 1 control, ideal for rapid exploratory evaluations within 17 minutes. And lastly, a full temperature range, used for the real-time monitoring of the deamidation process for an array of samples, ideal for developability assessment. This direct method provides the secondary structure location and the number of asparagine and glutamine residues that deamidated during a 2.5 hour period for an array of 21 preclinical candidate proteins and 2 controls, ideal for lead candidate selection. The following workflow simulates a design of experiment examining 4 samples against a control, while subjecting it to a thermal ramp starting at 25 to 65 degrees Celsius and collecting hyperspectral images every 10 degrees. Within the 1,800 to 1,000 wavenumber spectral region. Each of the resulting hyperspectral image is comprised of 223,000 spectra, and is the reason why the signal-to-noise ratio is significantly enhanced. This real-time hyperspectral image acquisition method is performed under accurate thermal control for a multiplex of samples using an innovative slide cell array, with a fixed path length allowing for comparability assessment of the intact therapeutic protein, which streamlines the workflow and limits variability in the results. The slide cell array has a designated area for background collection to be used to ratio against the sample to generate the absorbance spectra used to yield the hyperspectral image of each well, containing only 1 microliter of sample. No tags or labels are used, and there is minimal to no sample preparation needed. Once the hyperspectral image is collected in one well, the stage will move to the next well and collect the follow-on hyperspectral image. This process is repeated until all the images in the slide cell array have been acquired for that set temperature. The system will wait for the next set temperature to reach equilibrium and continue to cycle until the system reaches the last temperature and all the intended hyperspectral images have been acquired. The real-time acquisition of the field of view allows for minimal fixed volume requirements and the assurance of the image captured to be representative of the distribution population of therapeutic proteins in their formulation. This snapshot in time, at a given temperature, allows for the true comprehensive comparability assessment of all the samples in the array. The hyperspectral image is stored and can be re-examined at a later time for the investigation of deviations in manufacturing, to establish control strategies, or simply available for further investigations. The robustness of the method is ensured through its automation using our centralized data management system, with architecture designed to be 21 CFR Part 11 compliant and ensure data integrity. Multiple attributes may be monitored due to the high sensitivity and selectivity of infrared microspectroscopy, which is expanded to an array of preclinical candidates in the form of the series of data cubes. The hyperspectral images allow for true multiple critical quality assessment of an array of proteins in their formulation condition. The analyst can activate the dedicated software suite, and a quantitative report will be generated within minutes of the request. The fixed path length allows for spectral subtraction of the initial temperature from all subsequent spectra within each data cube to generate the different spectral data and allow for one of several two-dimensional correlation algorithms to be applied based on the selection of the software suite. The quantitative results are accurate, reproducible, and statistically robust. A comprehensive analysis describing the relationship of multiple critical quality attributes can also be performed to de-risk preclinical candidate selection, accelerating speed to market and ensuring safety and efficacy for patients.